In this session, we will understand what is polymerase chain reaction, the PCR. The polymerase chain reaction is a laboratory technique that is used to amplify or copy the segment of DNA. It was discovered by Carey Mullis and was awarded the Nobel Prize in Chemistry in 1993. He was working on oligonucleotide synthesis and he wanted to develop a modified version of dideoxy sequencing procedure using oligonucleotides, DNA templates, and DNA polymerases. While thinking of the same, his modified sequencing experiments trying to establish if extension of oligonucleotide primer by DNA polymerase could be used to remove unwanted DNTPs in the solution. He realized that if two primers were involved and they served to enable extension of DNA template, the sequence would efficiently be duplicated. He had been writing computer programs that required reiterative loops and realized that sequential repetition of his copying reaction could provide many copies of DNA sequence. Some hasty checking of the figures confirmed that the exponential increase achieved was indeed 2n. Hence, the PCR was discovered. The general PCR methodology is, firstly, the DNA duplex was heat denatured to give a single stranded. Then, the two oligonucleotide primers were annealed to their complementary sequences on the target DNA. Then a polymerase was used to synthesize complementary strand from the template strand by primer extension. This cycle was repeated to obtain many copies of the DNA. The typical temperature profile would be denaturation at 95 degrees Celsius, primer annealing at 55 degrees Celsius, and extension by Taq polymerases at 72 degrees Celsius. The reaction would give 4 number copies at the first cycle, then 8 at the end of the second, followed by 16 and so on. Requirements for PCR: one is the primer and the second being the DNA polymerase. Primer. 2 primers were required, one for each strand of the duplex. The primers were synthesized as oligonucleotides and are added to the reaction in excess so that there is no shortage during the reaction process. In primer designing, many aspects were considered. One being the sequence of the primer. The sequence information can be derived from amino acid sequence data, in which case the genetic code degeneracy has to be considered. There were 2 approaches. One is by incorporating a mixture of bases at the orbital position, which represented as a small portion of the mixture. Second, by the base inosine, wherein the inosine base was incorporated as the third base in the degenerative codon. If the primer sequence is taken from an already determined DNA sequence, it may be from the same gene, from a different organism, or maybe from a cloned DNA that had been sequenced during previous experimental work. The primer length is also important. It should be long enough to ensure stable hybridization to the target sequence at required temperature. The primer must be long enough to ensure that it is a unique sequence in the genome from which a target DNA is taken. It is approximately of the length 20 to 30 nucleotides that would be sufficient. DNA polymerases. When a standard DNA polymerase that is a clinofragment. Clinofragment is when the 5'3' exonuclease activity is removed from the DNA polymerase. It is thermolabile and is inactivated in the denaturation process, and hence fresh enzyme has to be added at the end of each cycle of operation. And it always had wanted an operator to be present near the machine for the duration of the process and required a lot of enzyme. It was carried out at 37 degrees Celsius, and primers could bind to non-target region, generating high target of non-specific amplified product. To avoid all these, we use thermostable form of DNA polymerases, the TAC polymerases, purified from thermophilic bacterium, Thermus aquaticus. The use of TAC polymerases helped in automating the procedure. There was no need to add fresh polymerases after each cycle. The key feature of DNA polymerase include processivity, that is, the affinity for the template which determined the number of bases incorporated before dissociation, fidelity of incorporation, rate of synthesis, and half-life of the enzyme at the different temperature. In addition to the DNA primers and polymerases, the usual mix of the correct buffer composition and availability of 4 DNTPs is needed to ensure that the copying of the DNA strands is not stalled because of inactivation of the enzyme or lack of monomers. Automation of the PCR cycle of operation is achieved by using a programmable heating system known as a thermal cycler. It takes small microcentrifuge tubes, that is a 96 well plate, or glass capillaries can also be used, in which the reactants are placed. Thin wall tubes permit more rapid temperature change than standard tubes are placed. A final practical consideration in setting up a PCR protocol is generally housekeeping and manipulation of sample. As the technique is designed to amplify small amounts of DNA, even trace contaminants can ruin the experiment. Also, even with the aerosols created in pipetting reagents can lead to cross-contamination, so a good technique is essential. Accurate labeling of tubes is also needed, and quality control procedures are important, particularly where analysis is being carried out in medical or forensic applications. This is the general procedure for PCR. Now, understanding the RT-PCR, that is very commonly used term today. So, RT-PCR uses mRNA as template in common variant of basic PCR. It is the reverse transcriptase PCR. It is useful in determining low levels of gene expression by analyzing the PCR product of complementary DNA prepared from the mRNA transcript. A single mRNA molecule can be amplified, but this is unlikely to be achieved in practice. The process involves copying the mRNA using reverse transcriptase as in a standard cDNA synthesis. Oligo-DT primed synthesis is often used to generate the first strand cDNA. PCR primers are then used as normal, although the first few cycles may be biased in favor of copying the cDNA single-stranded product until enough copies of the second strand have been generated. This has no effect on the final outcome of the process. One of the most important use of RT-PCR is in determining the amount of mRNA in a sample. A differing but known amount of competitor RNA is added to a series of reactions and the target and competitor amplified using the same primer pairs. If the target and competitor products are of different size, they can be separated on a gel and the amount of target estimated by comparing with the amount of competitor product. When the two bands are of equal intensity, the amount of target sequence in the original sample is the same as the amount of competitor added. Thank you for watching the video. For detailed notes, please do click the link in the description below. If you like, please do share and subscribe to our channel.